Hello, my name is Trina Ganguly, and the date today is February 10th, 2023. And today I'll be presenting on mucosal immunity and, in specific, the mechanisms that control mucosal immunity. To begin with, the mucosal immune system provides the first line of defense against toxic agents that enter the body through the mucosal membrane. The mucosal immune system has three main functions, which are to protect the mucous membranes against colonization and invasion by potentially dangerous microbes that may be encountered. Next is to prevent the uptake of undegraded antigens, including foreign proteins derived from ingested food, airborne matter, and commensal microorganisms. And lastly, to prevent the development of potentially harmful immune responses to these antigens if they do reach the body interior. In other words, to prevent the development of potentially harmful immune responses to exhaustion as antigens when they do make it past the epithelial barrier. And mucosal immune regulation is activated and induced by various pathways utilizing a variety of biomolecules. And so in the next few slides, I'll be getting into some more of the mechanisms that control mucosal immunity. To begin with, toll-like receptors are a class of pattern recognition receptors that play a major role in the initiation of innate mucosal immune responses and some adaptive immune responses to microbial pathogens. They detect conserved pathogen-associated molecular patterns, also known as PAMPs, on a range of microbes, including viruses, leading to innate immune activation and orchestration of the adaptive immune response. Activation of the toll-like receptor signaling pathway leads to the expression of numerous genes regulating host defense, including inflammatory cytokines, chemokines, antigen-presenting molecules, and co-stimulatory molecules. In specific, toll-like receptors signal through the recruitment of specific adapter molecules, leading to the activation of the transcription factors NF, KB, and IRFs, which then dictate the outcome of innate immune responses. Next, we have node-like receptors, which are highly conserved cytosolic pattern recognition receptors that perform critical functions in surveying the intracellular environment for the presence of infection, noxious substances, and metabolic perturbations. And sensing of these danger signals by node-like receptors leads to the oligomerization into large macromolecular scaffolds and the rapid deployment of effector signaling cascades to restore homeostasis. And NLRs are essential in recognition of microbial and pathogen-associated molecular patterns, which are also known as MAMPs and PAMPs, and have the ability to initiate and support immune responses through the formation of inflammasomes and the activation of NFKB, IRF, and MAPK pathways. Functions such as enhancement of the MHC transcription and presentation implicate node-like receptors in adaptive immunity, and their roles in reproduction indicate a broader responsibility of this gene family than it has been previously known. And so the potency of NLRs in inducing immune defenses is vital for the host, but can also provide some serious issues when dysregulation or malfunction occurs. And lastly, RIG1-like receptors are protein sensors of the pathogen-associated molecular patterns, which are, as mentioned, PAMPs, in the form of viral double-stranded RNA, which is dsRNA, and their motifs to induce expression of type 1 interferons and other inflammatory cytokines during the early stage of viral infection. In other words, they are known as important protein initiators of the earliest immune responses to viral infections. And in general, there are three types of RIG1-like receptors, which are RIG1, which is the retinoic acid-inducible gene 1, MDA5, which is melanoma differentiation-associated protein 5, and LGP2, which is laboratory of genetics and physiology 2. However, for this presentation, I will be focusing on the RIG1 and the MDA5. RIG1 and the melanoma differentiation associated gene 5, which contain N-terminal caspase recruitment domains, are activated upon the detection of viral RNAs in the cytoplasm of virus-infected cells. And these activated RIG1-like receptors induce downstream signaling via their interactions with mitochondrial antiviral signaling proteins and activate the production of type 1 and 3 interferons and inflammatory cytokines. And so lastly, I'm going to talk about some future research directions regarding these PRRs. And so there's a lot of research that still needs to be done regarding both toll, all three toll node and RIG-like receptors. And the study of PRR-mediated pattern recognition mechanisms will help to explain and further the knowledge of the signaling pathways and mechanisms of diseases, and also provide new targets and methods for the treatment of diseases and promote the development of immunology and oncology research and further improvements in their conceptual knowledge. And so, for instance, it has been found that TLRs and NLRs and RLRs all show great potential in immunotherapy of solid organ cancers as well as hematological malignancies. And so similarly to being used for cancer treatments, these pattern recognition receptors may be beneficial in other diseases too. Furthermore, it would also be beneficial to evaluate the efficacy of PRR inhibitors combined with conventional drugs for anti-autoimmune diseases to see how the PRRs react with certain drugs. And here are all my references. Thank you.